Welcome to the Ash Cloud. I'm Ash Sweeting. Today we are joined by Professor Rod Mackey, a gut microbial ecologist from the University of Illinois. Rod is leading an international research team pursuing a different approach to mitigating livestock methane. Rather than just studying methane in isolation, the team is looking at the biomolecular mechanisms that lead to the precursors of methane, to open up broader opportunities for intervention. The team has recently been awarded a $2.5 million grant from the Foundation for Food and Agricultural Research through their Greener Cattle initiative.
Most of the work that's been done tries to study that just in isolation in terms of methane production only. But if you dig into the fundamental reasons behind methane production, that's based on two important fluxes. One is hydrogen production, and the other one is hydrogen utilization. And so our study is really mechanistic. It's not a large herd study, but it looks into small animals that are based on this low and high methane emitting phenotype. So it's critical. It's really important to understand that. So I think the other thing that people don't really understand is if you inhibit methanogenesis, what are you going to do with the hydrogen that's being that accumulates in that system?
This approach can benefit both farmers and the climate.
What we'd like to be able to do is to redirect that hydrogen from being a waste of energy. So currently that amounts to between 5 and 12% of the gross energy in the diet is released as methane. If we can recapture some of that in more useful end products of fermentation, things like propionate and butyrate, those would be extremely beneficial. And we don't want to waste that hydrogen. It's a very good energy source, and we can't afford to blow that off. If that happens, it'll be like a win-win situation. We'll have contributed to mitigating methane emissions, but we'll have also captured that hydrogen, redirected it, and put that into useful end products. That in the end, if that happens, farmers won't have any problem in terms of accepting those sorts of technologies.
Understanding the details is critical.
A lot of that sequencing, that's done with short read technology. And I don't think that's good enough because it doesn't resolve it down to strain level. And when we're talking about anything that relates to rumen fermentation or gut fermentation, it's all about strain variation. And that's all important. So it's not the species.
The team has a history of success.
This Greener Cattle Initiative grant, it didn't just happen. It was years in the making. And so I've always been interested in comparisons between, say, the rumen system and the human gut, the human colon. But I started working with the people in New Zealand, Graham Matwood and Sinead Leahy, and we had two very nice publications in 2022 that are still kind of fundamental and led to the development of this grant. And it was all about hydrogen production and hydrogen utilization and understanding those fundamental things that are happening.
There is still a lot of work to do.
Are these animals always low and high methane emitting? Or if you change the diet, does that shift? And those things haven't really been done because we don't really look at that sort of thing. So we need a lot more money. And I think we need to be able to work in a lot of countries where there are large numbers of cattle. And so that are, like India and Brazil, those are where most of the animals are. Most of those in extensive systems.
A broad approach is essential.
But we need, much better alternatives. And that's why there's breeding strategies, other things that may, help. But understanding what trays to breed for, that's more likely to come from this sort of study.
Rod, thank you very much for joining me today.
Well, it's an absolute pleasure.
So to start with, how does the approach of the team you're leading to mitigating livestock methane differ from other approaches, and why is that important?
Yep. Okay, so that's really a very important question. So people have known about methane production in the rumen ever since the rumen animal digestive physiology was studied. And so there's nothing new about methane production. And I think that most of the work that's been done tries to study that just in isolation in terms of methane production only. But if you dig into the fundamental reasons behind methane production, that's based on two important fluxes. One is hydrogen production, and the other one is hydrogen utilization. And it turns out that the best way of disposing of reducing equivalents in the rumen system is to make methane. And so methanogens are really, really effective in reducing CO2 with hydrogen. And so in the rumen situation, most of the methane comes from this hydrogenotrophic pathway of methanogenesis. There's some evidence that maybe there's another pathway, the methyllotrophic pathway, but the quantitative evidence for that is lacking. And so I think that most studies don't take into account hydrogen production and hydrogen utilization and the balance, the fluxes of these important critical intermediates in the system. And so most studies to date, they use large animal studies with inhibitors and just don't really dig into those fundamental mechanisms, what's happening in the black box. Or they use just omics type approaches, sequencing, and that's it's a good start, but that's not the answer. That's the beginning. And so our study is really, really mechanistic. It's not a large herd study, but it looks into small animals that are based on this low and high methane emitting phenotype and uses that as a basis to try and understand these mechanisms for hydrogen production and hydrogen utilization. So it's critical. It's really important to understand that. So I think the other thing that people don't really understand is if you inhibit methanogenesis, what are you going to do with the hydrogen that's being that accumulates in that system? There's something in our favor, and that is that hydrogen solubility is very, very low, and so you soon reach like a supersaturation, and that's why we start to see hydrogen being emitted, belched out of the rumen, eructated, and so that's a loss of energy. And so our plan is to look at those fluxes of not only hydrogen, but formate, another important intermediate in terms of hydrogen formation, things like lactate, succinate, other intermediates that are turning over in the rumen, and how they might influence these fluxes, direction of hydrogen flow. So I think in the end, what we'd like to be able to do is to redirect that hydrogen from being a waste of energy. So currently that amounts to between 5 and 12% of the gross energy in the diet is released as methane. If we can recapture some of that in more useful end products of fermentation, things like propionate and butyrate, those would be extremely beneficial. And we don't want to waste that hydrogen. It's a very good energy source, and we can't afford to blow that off. If that happens, it'll be like a win-win situation. We'll have contributed to mitigating methane emissions, but we'll have also captured that hydrogen, redirected it, and put that into useful end products that in the end, if that happens, farmers won't have any problem in terms of accepting those sorts of technologies. So I think we've lost sight of some of the things that are happening in between feeding an inhibitor in the mouth and rumen fermentation and methane emissions. And our job is to find out what's going on inside this very complex system, the rumen. And of course, the other side of that is that 90% of methane emissions from ruminant animals come from enteric fermentation, and it's not from manure. So manure is part of it, but it's not a major part. Once we bring in other greenhouse gases, then we start to have to think more about the protein side of it, nitrous oxide emissions, another very potent greenhouse gas forming greenhouse gas. Yeah.
Am I correct in my understanding that by understanding the actual dynamics or the kinetics of this metabolism in the rumen, you open up greater opportunities or greater number of intervention points? So it means you've got broader areas where you can intervene compared to what is frequently happening nowadays, which is basically people are just trying to put a plug in the leak and get rid of the methane.
Okay. Yeah, Ash, I think that's probably a good way of stating it. So there are many other places to to look at that problem, and so that's why the turnover, the fluxes, and trying to get this accurate hydrogen balance in the system. So currently we don't know. There's a part of that budget, that hydrogen balance budget, that's missing. We can't account for it, and so if we can't do that, the chances of being able to sustainably, reliably, and reproducibly reduce methane emissions are much lower. So it's really fundamental. It's very mechanistic, but it's really fundamental.
And as you said, most 99% of emissions globally come from enteric fermentation. So that's what's going on.
90, yeah.
90, sorry. And you've also got the fact that the vast majority of animals are actually grazing, be that in small herds in Asia or Africa, or the pastoral zones of South America and Australia, and the ranches here, the breeding ranches, the cow-calf ranches across North America. Does the better understanding of these dynamics help to find solutions that will work for these grazing animals where they're not handled and fed every day?
Yeah, that's a very important point. And so I think these basic approaches will be very, very useful. But it's true that many animals in extensive systems, pastoral systems, you don't see them, maybe once a year or twice a year. And so feed additives don't cut it. And that's why there's still an extreme interest in trying to get a methane vaccine and to be able to deploy that. Say when you bring cattle into dip for ticks and things like that, then you would be able to vaccinate. But at the moment, that approach has not yielded anything beyond increasing salivary antibodies, increasing in vitro methane. But when it comes to in vivo, mitigation, it's not working. And so there's potentially other targets, but it's still an important part of the whole arsenal, that how we're going to reach all these other animals. And so for developed countries and dairy, where you see animals, 2 or 3 times a day, we can easily do it with feed additives. And beef cattle that in feedlot, at least, you feed them twice a day. And that makes it much easier. But we need, much better alternatives. And that's why there's breeding strategies, other things that may, help. But understanding what trays to breed for, that's more likely to come from this sort of study.
I know you're very much at the beginning of the journey in this study, and I'm asking you to predict. I'm about to ask you to predict what you might see come out of it. But what do you think the interventions from the farmer's perspective may look like, or the possible ones that may be on the table in a number of years when it gets to that point of the study?
That's sort of hard to predict. But I think so. On a big scale, everything that relates to just feed efficiency, things that relate to animal health, fewer animals, all of those things are going to make an enormous contribution. So that's not an essential part of our program. But in the end, I think we'll have much better predictors for what to select for, how to manage diets, additions to diets, things like that might be useful. And so, just natural plant compounds, things that might be, present in the diet of animals that are browsing as well as grazing, when there's a lack of grazing, maybe as you have in Australian regions. So there's going to be, I don't think there's going to be a single type solution. I think it's going to be, many different ways of doing it.
You've put together a team that covers, the globe, essentially, from Europe to Australia, New Zealand, North America, the Middle East. And can you just go through and tell me a bit about the team, who's in the team, and the skills and knowledge and experience they bring to the team?
Yep. So maybe I should just preface that by saying that, this Greener Cattle Initiative grant, it didn't just happen. It was years in the making. And so I've always been interested in comparisons between, say, the rumen system and the human gut, the human colon. But I started working with the people in New Zealand, Graham Atwood and Sinead Leahy. And we had 2 very nice publications in 2022 that are still kind of fundamental and led to the development of this grant. And it was all about hydrogen production and hydrogen utilization and understanding those fundamental things that are happening. So when I wrote the proposal, I knew that we needed more than just my own expertise, but a lot of other people. And so we're very fortunate to get this, I think, a very powerful international research team to work on this. And so we divided those into 6 different research hubs. And so the lead institution, of course, is the University of Illinois. And I'm working with a guy called Josh McCann, who's a professor in ruminant nutrition. And we're a beef cattle center. And I think overall, for all of the hubs, we're going through sort of a process of trying to work off what's already known about low and high emitting phenotypes, to work with that inocular in vitro bioreactors, and to establish levels that give us graded increments in dissolved hydrogen concentrations. So those are the critical parameters to measure. So that would be Research Hub 1. So Research Hub 2, those are the people in New Zealand. So that's Graham Atwood, Bill Kelly, Peter Jansen. Sinead's in a more of an administrative type position, so she's not directly involved in this grant. They have just a wealth of experience. They've got the flocks of sheep that are high and low methane emitting phenotypes. They have a good collection of methanogens that most other people don't have. And they're not really going to be working with animals. That's a bit too expensive for them. And so they're going to be looking more at hydrogenases. And so if you know anything about hydrogen, what's the enzymatic machine that handles those? Those are hydrogenases. And theirs is a proposal that looks at inhibiting hydrogenases of all these different kinds. And so there's lots of exciting work that can come out of that. So which potential hydrogenase? Is it a membrane-bound, a vectorial hydrogenase that's moving in hydrogen, or is it one that's effluxing hydrogen? What about the electron bifurcating hydrogenases, the new discovery in terms of just, hydrogen economy, balance? So that's a very powerful approach. Then the next team would be the team in Canada. And so they were previously all based in Alberta. So Lelu Guan at the University of Alberta, and Tim McAllister, Stephanie Green at Ag Canada in Lethbridge. Lelu is now moving to University of British Columbia, and that's also a beef site. So they're a team that have a lot of experience in terms of large herd studies. They've done a lot of fundamental work in 3-NOP, 3-nitroxypropanol, as well as asparagopsis. And so they've already got a huge foundation for doing this work. So we can interface with that and use it to go on and do much more mechanistic type studies. So to input some of those other fundamental measurements that are not currently there, but need to be made to make a full understanding of the microbiology that they're measuring. So that would be Research Hub 3. Research Hub 4 is a European-based one. There are actually 2 investigators there. So one is Sharon Hughes at Queen's University in Belfast. And her approach is very much to do culturomics. So trying to get that big database of reference isolates that contribute just not only to the database, but to understanding growth physiology, and then selecting those maybe to make a synthetic rumen microbial community. And in the end, trying to get genetic systems that make those organisms tractable, in which we can ask the question about which organism, which specific pathway, and which specific gene are really responsible for some of these things that we're measuring. So really, really important. The other person that's in the European team is Phil Pope at Norwegian University of Life Sciences. And his is very much sort of an omics type platform, but I think it's a much improved platform. He has a specialization in proteomics. Most of us can't really do that. And the improved database, the reference database, as well as his expertise in advanced omics will be very important. And I can add in there that for the first time, we suddenly have genomes for rumen ciliate protozoa. They're responsible for at least 20 to 30% of the hydrogen that's produced and gets converted to methane. But they've been excluded in all of these previous things because we didn't have a genome. And those reads were unannotated. They didn't have a home, but now they do. And now there's additional, ciliate protozoal genomes that we can use. And so I think we can go back and just reinterpret those sequence databases we have and reassign, new information to existing databases. And I might just segue in there by saying that a lot of that sequencing, that's done with short read technology. And I don't think that's good enough because it doesn't resolve it down to strain level. And when we're talking about anything that relates to rumen fermentation or gut fermentation, it's all about strain variation. And that's all important. So it's not the species. With the new technologies, we'll be able to get down to that level of resolution, but we'll need high, long read, high accuracy, amplicon sequencing. And so it'll all shift. Costs will be higher, but it'll all shift towards PacBio and mini-iron type technologies. You can't compare a full-length 16S gene to one that's 300 or 400 base pairs. There's just not enough information in there. And the same goes for other genes in metagenomics and metatranscriptomic databases. Okay, so that's Phil Pope. That brings in that's Research Hub 4. And I guess they'd be mainly sort of a dairy kind of interest. And then the next group would be Itzik Mizrahi, who's in Israel. And that's probably one of the best labs at the moment for doing omics type research on dairy cattle. So it's another dairy hub. And he's very much involved in metabolomics and then in constructing synthetic microbial communities. One of the problems we've had with synthetic microbial communities is that it's very hard to cultivate ciliate protozoa in those systems. They don't last very long. So we have to be able to redo that and try and promote survivability of ciliate protozoa in these synthetic systems to make it really count in terms of a study model. Okay, and then the last research hub is from Australia. So that's Broadening in Australia. And that's Fiona Soulsby, Diane Overkirk, Afroclidi, Matthias Hess at Davis. And theirs is very much trying to use acetogens as probiotics, trying to redirect some of that hydrogen and CO2 into acetate that would be a more useful end product. Now, of course, that's been tried before, and it hasn't been successful, so the acetogens don't compete with methanogens. Their hydrogen thresholds are higher, and so they're not as effective. And those acetogens, they do lots of other things. They're not obligate acetogens. And so it's easier to make a living doing something else than trying to push CO2 and hydrogen into acetate. So it doesn't happen very readily. But the opportunity is if we have a rumen in which there's methane mitigation, we're not making as much methane, we're elevating the hydrogen, dissolved hydrogen concentrations, can acetogen probiotics work? And is it possible that there are some other organisms in there that can help us with these elevated levels of hydrogen, which we must maintain at a low level in order to drive, anaerobic fermentation. And I think there's good opportunity. And I think for our proposal, that was the only sort of commercial addition. And so I think it was a good addition. It wasn't part of our original proposal, but they requested that we include that, and I was happy to do that. So you were instrumental in the early part of the proposal, anyway.
Cheers, mate. Thank you. You mentioned a lot of technologies in that while explaining the team. Would have this type of research been possible 5, 10, 15 years ago, or are the technologies that are being used, things that are on that kind of cutting edge side of things?
And I don't think 10 to 15 years ago, I don't think we were at this point. I think that what's happened is that there was an era when rumen microbiology, we were really in the vanguard. We were leading the chase, but that was early on because we had anaerobic technique and other people didn't. We're still very good at anaerobic technique, but I think we've fallen off the pace in terms of these other sort of omics technologies. And first of all, the environmental microbiologists came in, but now it's very much the human microbiome. And of course, that's a huge amount of money, and it's a very attractive area for people to do research in, and for young PhDs, postdocs, to study in that area. So I think that we're going to get to that point again, where we'll get that reinvigoration, more people getting back into this particular field, and being able to use these traditional techniques and the much more modern omics technologies that are so computer dependent. And so it involves just a different set of skills that some more mature scientists like me don't have. That's a word that the Australians use, mature PhD. It's old guys.
Yeah, well, this is only going to be audio, but looking at the color of your hair and my beard, I don't think there's any confusion about which groups we fall into.
There you are.
You mentioned earlier that the big exciting thing about this research is the potential to capture that energy that's in the hydrogen. You said the 5 to 12% of feed energy within the animal, or some of that within the animal, so that you're solving a feed cost, feed use efficiency problem for the farmer, as well as addressing the environmental concerns. So that whole win-win just makes the challenges of adoption much more straightforward. In terms of reaching that potential, what's needed in terms of funding, resources, people with the right experience? Very broadly, what's needed to accelerate this whole process?
I think a lot. So I think you could say, from the beginning, what you need is a lot more money. So it might sound like the Greener Cattle Initiative is a big grant, but it's not. It's divided amongst all those different research hubs. And to have, even to maintain herds of low and high methane emitting sheep or cattle, it's very expensive. And so most people don't want to do that. We can use green cattle or the green feed system, and we can measure them. But to do studies, are these animals always low and high methane emitting, or if you change the diet, does that shift? And those things haven't really been done because we don't really look at that sort of thing. So we need a lot more money, and I think we need to be able to work in a lot of countries where there are large numbers of cattle. And so that are, like India and Brazil, those are where most of the animals are. Most of those in extensive systems. And so we need to think seriously about how to deploy these technologies and have really technologies that are relevant and applicable to those sorts of systems. And so that's going to be, that's like down the road, but it's something we need to start thinking about now. So with money comes, a lot more interest. And so for people in universities, we don't have a mandate to do anything. The mandate is to go and get a grant, and so you're looking for money constantly, and so I think that it's going to attract a lot more top labs into trying to compete for this funding, and competition's good, and so in the end of it, we'll get many more top labs. Think about how much money it took to do the human gut microbiome studies. it's enormous. I don't think we'll ever get to that level of funding, but we need to get somewhere much more than we have now. So it's funding. Then it's the much improved like intellectual contributions to trying to study and find new ways. And chemical engineers know a lot about gas solubilities, fluxes, dynamics. Can we measure hydrogen turnover with new technologies? Yeah, proton NMR and other techniques like that. But we need chemists and other people who have these skills. It might be easy for them, but we need to be able to embrace and capture this type of knowledge. Better researchers, and then that translates into more PhD students. So funding for PhD students, funding for postdocs who want to go into this area of research, and so that's really important. And then there's all the facilities that go with it. Animal facilities, holding for those herds of animals. Animal studies are just exorbitant. So are human studies, but animal studies are very expensive, and that's why it's easier to do like bioreactor type studies and replicate at the bioreactor stage instead of having 1,000 or 2,000 head of cattle. So all of those things are going to be really important, and I think that people are aware that to get this whole thing moving and rumbling in the right direction, it's going to involve all of those sorts of things.
Excellent. Thank you very much for all your wisdom and sharing with us what this grant and this exciting team. Before we go, is there anything else that you'd like to add that we hadn't discussed?
I think we've discussed most of it. I think the idea of gas solubilities is really important. And so that's the effective thing. Breath methane is one thing, but measuring dissolved gases is another. And so the only gas that's really soluble is CO2. And actually dissolved CO2 is also important to the whole animal physiology part of it. I think that, we need to integrate the rumen stuff with the animal physiology a lot better. So it's not only about the microbiome. In the end, it'll be about host microbe interactions. And then another thing that I've been pushing is this idea of, it's fine to say that methane is a culprit for a lot of the greenhouse gas forcing, but it turns out that nitrous oxide is 298 times more than CO2, and then you've got the half-lives to factor in. But the half-lives favor us. I think if you take a half-life of methane of 10 to 12 years, means we can make an immediate contribution, whereas to ultimately control, global warming, it's going to involve a lot more, and people will have to stop producing as much CO2. And until that happens, we're still going to be in a bad place. So what happens when animal production goes up and we exceed our sort of targets? That's something for the future, but we have to feed the planet. It's really a very exciting time. I think I'm energized and very excited to be at this time. Yeah.
Rod, thank you very, very much for your time.
Yep. No, it's a pleasure. Thanks.
You've been listening to The Ash Cloud with me, Ash Sweeting, in conversation with Rod Mackey, recorded in California in October 2023.